All right, it is 12:30, so we will get started for today. And what we are going over today is we're going to finish up our discussion on the citric acid cycle. So we have a little bit to talk about the regulation. Then we're going to look at the other uses of the citric acid cycle other than for energy. What can the intermediates be used for? From there, we're going to switch over to a new chapter where we talk about the electron transport chain. So we're going to introduce the mitochondria, look at the important parts of the mitochondria, then start talking about the components of the electron transport chain. But on Tuesday, I said where we would start is just a little review. So this was the slide that we ended on. And so I just have some questions. So it allows you to go back and look at these slides, make sure we understand everything and there's no confusion. So I have a bunch of questions of 1, 2, 3, 4, 5. And I just have them in one poll. All of these questions, and I know I just skipped question 4, so we'll get back to that, but let's go to question 5, a little bit out of order here, where I have a poll and we're looking at a bunch of things. So let me just get that first. Okay. So in this poll, I'm asking for 1, 2, 3, 4, 4 different substrates. I want you to tell me what happens when we increase the concentration of these substrates. Will the citric acid cycle flux increase? Will the flux decrease? Or will there be no change in the flux? So take a minute or so, review what we talked about on Tuesday, and see if you can come to some consensus on what's going to happen with the flux here. All right, so hopefully you had some time to review what we talked about, and let's go over this one by one. So let me stop this poll. All right, so this image on the right is going to be our guide, right? So this is telling us how different things affect the flux of citric acid cycle. So what citrate will do is that it will go and inhibit citrate synthase. And citrate synthase is one of the enzymes responsible for controlling flux. So the more citrate you have, the slower the citric acid cycle will actually go, because it will go and regulate itself. So increasing concentrations of citrate, lower flux of citric acid cycle. ATP down here, ATP goes and inhibits, you just follow the line here, isocitrate dehydrogenase. Again, isocitrate dehydrogenase is another one of those enzymes that is far from equilibrium. So it is one that can also control flux. So it will slow down this enzyme, which ultimately slows down the citric acid cycle. Succinyl coenzyme A, again, it has the same type of mechanism as citrate did. With increasing amounts of succinyl coenzyme A, it will go and inhibit the enzyme that creates it, feedback inhibition. And the enzyme, again, is alpha ketoglutarate dehydrogenase, which is very negative, delta G. So it's another enzyme that works far from equilibrium. So it's another control point. And also, succinyl coenzyme A will go and inhibit citrate synthase. So it's inhibiting two enzymes when you have a large concentration of it. So it will also decrease the speed of the citric acid cycle. G6P, glucose 6-phosphate, which is the first product of glycolysis, that itself has no effect. G6P doesn't go and interact anything with the citric acid cycle directly. So on itself, it doesn't affect anything. But if you have a lot of G6P, that can run through glycolysis, create a lot of ATP, eventually create a lot of citrate. So eventually, it will have the effect of slowing it down, but just by itself, it doesn't do anything. Now, thinking about how different metabolites can affect flux of our processes, I do have one question here that ties both glycolysis and citric acid cycle together. So jumping back to question 4 here. Why would it be advantageous for citrate to go and inhibit PFK? And remember, PFK is the major control point for glycolysis. So if citrate does a lot of things, and something else it does is it goes and inhibits PFK. Remember that straight up and down line means like inhibit when you see that in different biomolecular flow charts. So can anyone think of the logical reason why it would be a good thing for citrate to inhibit PFK? High glucose levels. So if there is citrate, there would probably have meant that there was high glucose levels. That is correct. Thinking on the grand scheme of the cell, though, and the different needs pulling at the cell with its limited resources, how can we connect that idea of that there's high glucose levels, therefore there was a lot of citrate, and then we go inhibit PFK? Why is that advantageous, I guess, is what I'm trying to drive at. The cell already has enough energy, no need to continue glycolysis since PFK is a major control point. Yeah, that's basically the logic here, right? So PFK is the control, is flux for glycolysis. Glycolysis makes ATP, and it does it by running the citric acid cycle. It makes pyruvate. That pyruvate goes through the citric acid cycle. The first product you make for the citric acid cycle, which is, by the way, this is mitochondria. Why glycolysis is cytoplasm. So the first product you make is citrate. If you have a lot of citrate, what will happen is that it will start being transported out of the mitochondria into the cytoplasm. And if you have such a large amount of citrate for this to happen, this means you have a lot of ATP that's going to be available. So thinking as a cell with very limited resources, you don't want to keep spending your glucose to make ATP when you have this much citrate, because you have ATP. So stop glycolysis. And the way to stop glycolysis is inhibiting PFK. So citrate goes and inhibits PFK. You can use that glucose for other things. Maybe start running the pentose phosphate pathway, right? So that's before PFK, and you can just shuttle off over there. Or how about you start making glycogen, right? Who knows when you're going to eat next? Start storing that instead. But yeah, that's why, it's a pretty smart system where something further down the line, a product that's created like one of the last steps, can go and inhibit the very first product. So you don't have to waste your resources creating something you don't need. That makes sense? Or any questions concerning that idea? All right, if not, we can continue to move on. So one more question. In the citric acid cycle, we never actually see oxygen being used. However, it is known that the citric acid cycle will only run if oxygen is present. Therefore, how is this system set up? How can this system sense oxygen is around if none of the reactions we looked at ever had any oxygen in it? And why would such a system like this be in place? But let's think about the how, because does anyone have any good ideas of how this system can be regulated by oxygen without us ever actually seeing oxygen being involved in the mechanisms we looked at on Tuesday? And there's one key molecule. One key molecule. Since oxygen is the final electron acceptor, if it's gone, NADH, FADH cannot get rid of electrons, and no more NAD plus FAD is made. Yep. That is the logic. Right. Remember we talked about that one of the very last things we talked about on Tuesday was this idea that NADH is one of the major control points for the citric acid cycle. You can see if you have a lot of NADH, it will go and stop the 3 major enzymes. It will also stop the PDH complex, pyruvate dehydrogenase complex. So under conditions of oxygen, NADH, and we'll look at this today, gives electrons to oxygen. I guess I shouldn't draw the arrow, but so it gives electrons to oxygen and it becomes NAD plus, and this cycle can continue to run. If you don't have oxygen, this doesn't happen. The concentration of NADH increases because you have no way to get rid of it, and it will go and stop all the critical enzymes. So this is how the citric acid cycle can run as like sensing oxygen, where we don't have anything that actually senses oxygen per se, but we have a molecule that can interact with oxygen, and the levels of NADH is what controls citric acid cycle the greatest. Well, even if someone else has put in the answer by 1.5 seconds, feel free to still enter it in. You never know. You might have something else that's like sounds the same, but coming at it from a slightly different angle or something else to add into. So put it in. It's always good to see different people might have slightly different perspectives. Right. And that is all for the PowerPoint we had on Tuesday. So let's go to today's PowerPoint, which I should have put up this morning. Yeah. And we just have a few more things to talk about the citric acid cycle in terms of what else it can do before we move on to the electron transport chain. And we're going to talk about the amphibolic nature of the citric acid cycle. That is, it can do both anabolic and catabolic reactions. It's not only used to build stuff, but things can be destroyed to enter the citric acid cycle. So let's talk about the Anaplerotic reactions. So these are reactions that will drain the intermediates away from the citric acid cycle for other things. One of them we already talked about is making glucose. So we looked at this mechanism last week, Thursday, where we saw oxaloacetate going to malate, going to glucose. So this oxaloacetate no longer makes ATP because it's getting just taken away to make glucose. Along those lines, oxaloacetate can also be used to make amino acids. And we actually did look at this very briefly when we talked about how oxaloacetate makes glucose. Because if you remember, and we didn't talk about it all that much, so no problem if it doesn't sound that familiar to you. But when it comes to making glucose, the problem with oxaloacetate is that it's in the mitochondria, like everything in the citric acid cycle is. And all the enzymes that make glucose are in the cytoplasm. So to get it out of the mitochondria, we have to convert it to malate because malate has a transporter, while oxaloacetate doesn't. So we can transport malate out of the cell. Well, to do that, we actually can do it a different way where we have oxaloacetate goes to aspartic acid. So we convert oxaloacetate to asp instead using alpha-ketoglutarate. So that's one way we can use oxaloacetate to make amino acids. We can actually make aspartic acid through oxaloacetate. And we looked at the transporter. I didn't really mention it all that much, so it's not a detail I'm too worried about that, just mainly focus on the oxaloacetate to malate to glucose idea. Big thing that we're going to look at after we talk about the electron transport chain is the creation and destruction of fatty acids. And what we'll see is that acetyl coenzyme A is used to build fatty acids. However, like oxaloacetate, we can't really just transport acetyl coenzyme A from the mitochondria to the cytoplasm. So we do the step of creating citrate. As we talked about like 4 minutes ago now, citrate can leave the mitochondria. And one of the things citrate does is make fatty acids. Again, the logic of that being, if you have a lot of citrate around, you have a lot of energy, no need to break it down, let's store it, right? So we're going to use it to block PFK, and we're going to use it to make fatty acids or cholesterol. And alpha-ketoglutarate, just mentioned it with oxaloacetate, but alpha-ketoglutarate is really a good starting material to make your amino acids. And alpha-ketoglutarate is a big ammonia sink as well. So you take your ammonia, you attach it where the C double bond O is, you remove that C double bond O to make glutamate, and glutamate can be used to make a lot of different amino acids. Succinyl-CoA to porphyrin rings, so that's another one. So anything with the red arrows, that's the catapleretic reactions. So that's reactions that drain the fatty acid intermediates to make other things. So a question for people, another one of these, critical thinking, you are now almost ready to leave college type of questions. So blood glucose, when that's high and the cell energies needs are met, insulin will come in and say, start storing that glucose as glycogen. Now, insulin can also promote the conversion of pyruvate to acetyl acid coenzyme A. And as we saw, acetyl acid coenzyme A is the first metabolite needed for the citric acid cycle. So do you think it's counterproductive for insulin to say, okay, stop using glucose for glycolysis and start storing it instead? And at the same time, start breaking down pyruvate to acetyl coenzyme A. You want to have thoughts on like why the cell might have such a system laid out for itself. Why does insulin do these two things? What is the logic of breaking down this pyruvate to acetyl coenzyme A? If we're already making glycogen, and we saw pyruvate can be turned into oxaloacetate instead to make glycogen as well. Yep. So both of you got that correct. It's because, and this is a concept that also goes back to this is a concept that goes back to biochemistry 1, right? Glycogen is made out of glucose. Glucose is made out of a bunch of carbons and OH groups. For every, I forget exactly what it is, but it's basically for every 1 gram of glycogen you have, you need to store 2 grams of water, because water is just attracted to glycogen a lot. Glycogen is completely hydrated. So storing glycogen, you have to carry on a lot of weight. So it's not that efficient. It's good for emergency in like your muscles, and you need it in your liver because your brain needs glucose. But as a overall energy storage idea, it's not great. However, fatty acids are a great energy storage mechanism because they're hydrophobic. They don't have any water with them. So in your fat cells, there's nothing but fatty acids. So you don't have to carry around extra weight. So the idea here is the acetyl-CoA, the pyruvate, can be stored in a better storage system. We can take some of that and just say, you're going to be a lipid now because it takes less energy to carry around a lipid than it does a glycogen molecule. And also, as we just looked at, it's possible that this acetyl-CoA will be run to different parts of the intermediates and start creating different things. So you can start creating amino acids now. You can start creating your porphyrin rings. But yeah, it's mainly because, well, we're not running the citric acid cycle for energy, where we just want the intermediates. The intermediates can also do things. Now, one more thing here. So thinking about the citric acid cycle, thinking about glycolysis and gluconeogenesis. So animals, we cannot take acetyl-CoA and make glucose from it. Because remember, to make glucose, we start with pyruvate, right? And going to pyruvate to acetyl-CoA is a one-way street. You cannot go backwards. PDH can never run backwards. However, if you give a rat carbon-14, and what carbon-14 is, it's radioactive carbon that you can measure, right? So if you give it radioactive carbon in acetate, so you just give them acetate, which is the acetyl part of acetyl-CoA, what happens is that you extract glycogen from them, and now they have C14 glycogen in their muscles. How is that possible? From malate. That is correct. Yeah. So acetate, right, can be converted into acetyl-CoA. From that, we run the citric acid cycle until you get to malate or oxaloacetate. And you'll still remember, when we run the citric acid cycle, right? So there's the citric acid cycle. And oxaloacetate is here. Acetyl-CoA is here. That forms together and makes citrate. Now, we lose 2 carbon dioxides along the way. They're gone forever from your body. You breathe them out. Now, the C14 that we're talking about is coming in on this step. So C14 is here, and it can be fed in. The CO2 It's from our original oxaloacetate. So we lose a CO2 from here, we lose a CO2 from here, but we still have C14. That's not lost yet. So that theoretically can go all the way till you hit malate or oxaloacetate, either one. Once you're in the malate, you can be transported out of the mitochondria into the cytoplasm. You remake pyruvate. You go all the way back to glucose to make glycogen. All right, and now, even though technically acetyl-CoA isn't directly made into pyruvate, by going around the cycle and just getting lucky by some of that malate leaving, you can have your labeled glycogen. So again, this just shows you that everything we're talking about this semester, for the most part, it's all interconnected. And I think at the very first lecture, I showed you like that giant map of all the lines, everything just crisscrossing. Yeah, everything in the cell when it comes to metabolism, it's just like they're connected. You can enter at any point and go through enough cycles to end up like at anywhere. And it's all very much controlled based on what the cell needs at any given time. So any questions about that idea? All right, so we can move on. Now let's talk about the anaplerotic reactions. And these are the things that make citric acid cycle intermediates. We already talked about pyruvate and pyruvate carboxylase. Pyruvate carboxylase is the first step in gluconeogenesis that makes oxaloacetate. So you can run this without, having to use it to make glucose. The oxaloacetate can be fed right into citric acid cycle and be an intermediate there. Of course, the normal way for pyruvate to go in is through PDH, pyruvate dehydrogenase, and make acetyl-CoA. What else can happen is that pyruvate can be made into alanine. And we briefly talked about this when we looked at, glycolysis without oxygen, when it can go to lactate. We briefly mentioned that pyruvate can go to alanine as well. So through a transaminase reaction, and if you remember, a transaminase, so amines is in there, you're moving an NH3 group. So we move an NH3 from glutamate to pyruvate, and that makes our friend alanine. By doing this, we will create alpha ketoglutarate from that glutamate. AKG can go right into the citric acid cycle. This is all very much controlled. So acetyl-CoA can actually activate the process of going from pyruvate to oxaloacetate. And again, we talked about this, I believe, when we talked about gluconeogenesis. If you have a lot of acetyl-CoA, that means you have a lot of energy. So that pyruvate is going to be made into oxaloacetate for storage purposes. But it could actually still run through the cycle. One thing you would want to do this, or sorry, that did not make any sense. Why would you want to increase the intermediates of the citric acid cycle? Flux, right? If you take that pyruvate and instead of going to acetyl-CoA every single time, you just start putting into oxaloacetate or alpha ketoglutarate, or you start breaking down amino acids to go into the citric acid cycle, you're going to speed up this whole process 60 to 100 times. So during exercise, you're going to start breaking down anything that can go into the citric acid cycle and start throwing it in there to create energy. And I mean, this is also kind of seen like when you start fasting. One of the first things to be broken down and thrown into the citric acid cycle are your muscles. Start breaking down that protein. You can make that as intermediates. Plus, muscles take up a lot of energy themselves, so it's a way to save energy. Yeah, and overall, like I said, by increasing these intermediates, you are just speeding up how fast you can make ATP. So that is basically all I have to say on the citric acid cycle at this point. So if we just take a moment and bask in the glory of all we've learned this semester, we've learned how to take glucose and all the way through the citric acid cycle. And we also learned about all the side pathways, pentose phosphate, the different fates of pyruvate. And all through this, we've been making this little pesky molecule NADH and occasionally FADH2. We are finally at the point where we can talk about how we get all of our energy back from these molecules. And that is the electron transport chain, which is just a bunch of proteins in the mitochondria that do nothing but pump protons and transport electrons through each other. And so basically what we're going to look at next is NADH and FADH2 giving off their electrons and to be remade into NAD plus and FAD. These electrons are just passed from one redox center to another in a very ordered manner. And you keep passing it to something that wants it more and more and more, with your final electron acceptor being oxygen, which we will convert into water. Every time we're passing these electrons, since we're passing them downhill, that is, we're doing favorable reactions, we can do unfavorable reaction of pumping protons against the gradient. This creates a proton differential, a pH gradient, across the inner mitochondrial membrane. Then we can allow the protons to flow through the gradient naturally through an enzyme called ATP synthase, which is like a giant turbine, almost like a giant wind turbine. And we can use that spontaneous process to do the non-spontaneous reaction of making ATP. So that's what we're going to look at in detail today and most next week as well. Time to get into the nitty-gritty here. First, let's talk about our friend mitochondria. This is where all of our oxidative metabolism is going to happen. Contains such favorite enzymes as pyruvate dehydrogenase, all of your citric acid enzymes, stuff to do fatty acid breakdown synthesis, all your electron transport chain enzymes, and other oxphos enzymes. And the mitochondria itself is made out of two membranes, right? The outer membrane and the inner membrane. And between the inner and outer membrane, we have a space that we call the intermembrane space. So the intermembrane is between outer and inner. Now, the inner membrane itself can be invaginated, and these special like little chasms in the invagination is called cristea. Right, so it's technically connected to the intermembrane space, the invaginations, but they are different chemically. And the number of cristea really relates to what cell the mitochondria is in. Inside the inner membrane is what is called the matrix. And it's shown here. It's like a gel. And inside this gel are all the enzymes that are needed for oxidative metabolism, any substrates, any cofactors, and a bunch of inorganic ions. So this is where a lot of the oxidative phosphorylation is going to happen. Your matrix also contains your DNA, which is, if you remember from biology, is circular. Again, one of the reasons why we think ancient eukaryotic cells just ate a mitochondria and just lived with it is because, we have linear DNA, the mitochondria has circular DNA. Mitochondria has its own RNA, of course, and ribosomes. But what is interesting is that the mitochondria is so dependent on the host cell that out of the 1,500 proteins that it needs to survive, it only makes 13. And the logic that people have come up with this is that these 13 proteins are so hydrophobic that there is no way to create them in the cell and transport them into the mitochondria without something going wrong. So the mitochondria still has to make 13 of its other proteins. The other 1,487 proteins are made inside the cytoplasm, and the cell is transported to the mitochondria. So that's our overview of the mitochondria. So any questions about the information on this slide or anything up to this point? All right. So the crustea can be simple, just like tubes, or it can be very complicated structure. Again, that depends on what cell types you're looking at. And one of the reasons why we have these invaginations is, like I said, they're micro compartments where we can have different concentration of substrates and ions when compared to the intermembrane space. And this is really where we're going to do our proton pumping. And so you have a large pH gradient in the crustea. And we can control that because they're in this little chasm. They don't really have anywhere to diffuse. You only have one entry point into the intermembrane space. So even if they diffuse out, they're not going to be diffusing fast. So this strategy of having this little chasm, keeping this pH gradient stable, is going to really speed up oxphos for us. By the way, anytime I say oxphos, that's just me being lazy for oxidative phosphorylation. So just a simple question here to make sure we're still with us. If you had 2-cell mitochondria that you're looking at, which one do you think would have more crustea? Do you think a heart mitochondria would, or do you think the liver mitochondria would? All right, so the answer for this would be the heart. Remember, so I just said that these crustea is what is keeping our pH gradient. So the more of these you have, the more oxidative phosphorylation reactions you can run, the more pH gradients you have. The pH gradients are used to make ATP. The heart's job is like a giant muscle. It is a giant muscle. So it uses ATP all the time. So you need mitochondria that can make a lot of ATP for your heart. So that's why you would expect it to have more crustacea than the liver, which isn't a muscle. All right, so let's focus in now, and we're looking at the image that you're looking on your left. The blue is the outer membrane, the yellow is the inner membrane, and labeled on it is the space between the two membranes, which is the intermembrane space. Now, the outer membrane of the mitochondria is really porous. It has these proteins called porins. I'm not sure if we talked about porins in biochemistry 1 or not, depending on what semester you took it. But what a porin is, it's an enzyme, or a protein rather, in the membrane that allows stuff to go through it. It's like a giant hole that you can control. And the outer membrane is just so porous, anything that's less than 10 kilodaltons can just go in, no problem. What this means is that the cytoplasm in inner membrane space is basically the same. There's no difference between them in terms of metabolites and ions. By natural diffusion, it all becomes the same. While the inner membrane has a lot more transport proteins, it's very, very selective on what goes into the matrix. And the only thing that can go through by itself are the molecules oxygen, carbon dioxide, and water. Everything else, the transport is controlled by proteins in the inner membrane. So we have different transports for ATP, ADP, pyruvate, calcium, phosphate, as we looked at citrate, malate. There's just a lot of them. And the reason why we control this is we want to control metabolism, right? We want to control the amount of substrates. More importantly, we want to control the ion gradient. We want to make sure that protons are only traveling through the proteins we want them to travel through. So it's very closed off. It's very controlled. And this allows us to have the mitochondrial matrix be completely different than the cytoplasm. They're like two completely different environments by having the inner membrane constructed the way that it is. So any questions about that or anything up to this point? All right. Now, since the inner membrane is so regulated, we need various shuttles. Now, this picture, hopefully it looks familiar to you. This is an image we looked at for getting oxaloacetate out of the mitochondria matrix and into the cytoplasm for gluconeogenesis. This is also used to transport NADH from the cytoplasm into the mitochondria. So remember, in glycolysis, we make some NADH, and oxidative phosphorylation happens in the matrix. So this needs to be transported into the matrix. And to do that, we have two different systems we're going to look at. This system, malate aspartate shuttle, like I said, we looked at it. Here, the picture is still showing if you want an oxaloacetate out of the matrix to the cytoplasm, this can run in reverse. So let me just draw the correct reverse arrows. So the idea here is that you take oxaloacetate, and usually you're going to make it into malate. And when you do that, NADH in the cytosol is converted to NAD+. Malate can be transported, turned back into oxaloacetate. And when you do that, you convert NAD plus in the mitochondria to NADH. So in essence, you are transporting one equivalent of NADH from the cytosol to the mitochondria through the intermediate of oxaloacetate and malate. You can also go the aspartate route, but this is not as frequently used as the malate route. So that's one type of shuttle. That's the malate aspartate shuttle. So this slide is mislabeled. It's not the glycophosphate shuttle. The next slide is the glycerophosphate shuttle. So what the glycerophosphate shuttle is the major way NADH is transported into the mitochondria. And the amount that this shuttle is present in different organisms really is variable. The most it's been found in is insect flight muscles, which makes sense. These insect flight muscles, by the way, are the cells that produce the most power out of like any cells scientists have ever looked at or any tissues. Flight muscles of insects are the biggest power generating muscles and all known on Earth that humans have discovered. So let's take a look at how this works. So the enzyme that's mainly used here to begin with is 3-phosphoglycerol dehydrogenase. Hey, look, it's our friend dehydrogenase. Hopefully 20 years from now, if you randomly come across the word dehydrogenase, you're going to remember every time I pointed it out and said, look, there's NADH involved. So here we're taking DHAP. Hey, it's our friend DHAP. The more you learn about metabolism, the more the same players keep showing up. So we take DHAP and convert it into 3-PG, 3-phosphoglycerol. And doing that, NADH goes to NAD+. Then we can take that 3-phosphoglycerol, turn it back into DHAP through a protein called flavoprotein dehydrogenase. This is an enzyme that's actually found in the inner mitochondrial membrane. That FADH2 can then give electrons to electron transport chain, which anytime I say the word ETC, that doesn't mean etc. That means electron transport chain. So doing this shuttle, just flip-flopping between DHAP and 3-PG, we can transport NADH, move those electrons into the mitochondrial membrane by having our intermediate flavoprotein dehydrogenase. So any questions about how this shuttle is run or anything at all up to this point? All right. If you do have questions, feel free to continue to type them out. So here is our big overview of ETC. And we're going to look at 4 different proteins, and well, they're actually complexes, 4 different complexes that are made out of multiple proteins. The whole idea of the electron transport chain is that we start with either NADH or FADH2. Those are our original carriers, and we give their electrons to these complexes, complex 1, 2, 3, or 4. These complexes then gives these electrons to carriers, coenzyme Q or cytochrome C, depending where you are. And also shown here, Q, coenzyme Q or cytochrome C. These enzymes, or these carriers, they're not enzymes, these carriers move electrons to different complexes. They're like delivery men. Until finally you get to complex 4, which we use to create ATP. Complex 1 and 3, there is what they're pumping out the hydrogens into our crystea, right? Into our intermembrane space. And then the ATP, complex 4, allows it to come, or sorry, ATP synthase is actually down here. ATP synthase is what allows it to come back in. The way that these were discovered is through a lot of experiments where we use different inhibitors to block the different complexes. So if you ever wondered why cyanide is deadly, it's because it blocks complex 4. You can't make ATP and you die. Rotenone, amytal, antimycin A, all of these block the different complexes, and that's how we found out they existed. These complexes themselves, they're in the membranes and they can move in the membranes. They're not like solid, right? Remember the membranes, remember from Biochem 1, it's like it's a sea of fat, I guess is the best way to put it. Those lipids are not frozen in place. They're liquid / solid. So these things can float on that sea of fat, or sea of lipids, if you will, as like big islands. And they usually associate together into giant complexes. So like this 3-2-4-2 means you could have just like 2 3s and 2 4s traveling together as a unit, or 1 1 3 2 4 means you have a 1 2 3s and 4 just traveling together as a unit. So these things often come together and move around in your lipid membrane, or your inner membrane rather. And this whole process is exergonic, which makes sense because we're making ATP, which is an endergonic reaction. So to make something unfavorable, you have to do something favorable. And we can calculate the energy, how much energy is given up, or rather how much energy is produced through this, using everyone's favorite equation, the standard reduction potential equation, where delta E, if you remember, is E acceptor minus E donor. And here is our half reactions for this process as a whole. NADH and oxygen making water. Here's our standard reduction potentials. Now here it's going to be oxygen is our acceptor, right? It's our final electron acceptor, where NADH is our donor. So to calculate the reduction, the delta E not prime, it's acceptor, so oxygen, minus the donor. And overall, 1 molecule of NADH will produce roughly 1.130 volts of energy. We can convert this into kilojoules per mole using delta G equals minus NF delta E. Remember, N is number of electrons. F is Faraday's constant. Delta E is what we just calculated. Overall, the free energy of this is minus 285 kilojoules per mole. You break that down into how much ATP you can make, and theoretically, you can make 6 ATP from 1 NADH. In reality, we can only make 2.5. Can anyone think of the reason why we don't make 6 ATP for 1 NADH? We only make roughly 2.5. What would be that reason? Well, I and take a much needed water break. Is it because the H are transferred one at a time? No, I mean, I don't think that's it. Because theoretically you could transfer more H's, I guess, but it could be it. Like it could be that there's a restriction on how many hydrogens you could pump. So I didn't think about that. So it's possible it's that. What I was thinking is that energy transfer is never 100% of what you calculate. Energy is always lost. So it can be lost as like vibrations of proteins, which we'll actually see is what happens in complex 1. When we're transferring electrons, some of that energy is used to change protein conformation. It can also be lost as heat. So you're just losing energy along the process. So you can't just harvest all this energy to create ATP at the same time. Yeah, kinetic energy, right? All of this is potential energy, but it's never 100% like captured. You're always going to lose some of it somewhere along the line. Be that in a car engine or the cell. I think the car engine's only like 20% efficient. So at least the cell's better than like an engine. So here, since we're probably going to ask another question like this, if not on test 2, definitely the final. Let's remind ourselves how to do these calculations then. So some prokaryotes who are anaerobic use nitrate instead of oxygen. So let's just say these organisms are 100% efficient. How many ATP can you make by taking NADH and oxidizing by nitrate? So the oxidation of NADH by nitrate. Keep in mind that to create 1 ATP, you need 30.5 kilojoules of energy, while the reduction potential of NO3 is 0.42. So let's see if you can do that calculation. How many ATPs can you theoretically make by using nitrate instead of oxygen? Whoops, sorry. So take some minutes and try to work through that math. And I have the example, I have the oxygen problem up there for you to look at and use as a guide. And I guess you also need Faraday's constant. It would be helpful if you had that. So Faraday's constant, 96485. Oh, the units are like kilojoules per mole volts. All right, so hopefully you had at least got somewhere with that. Let's take a look at this. So the only real difference between this calculation and the calculation that I show you here for oxygen is what the acceptor is. So instead of the oxygen 0.815, we use 0.42 for our nitrate. And the difference in voltage then, or the voltage potential, is 0.735. Then we plug it into our delta G. Delta G equals -2. The 2 comes from we're moving 2 electrons. Faraday's constant times our voltage, 0.735. That equals a negative 142 kilojoules per mole, which if we take that divided by 30.5, then we can roughly make 4.6 ATP, which just shows you the power of oxygen, right? Oxygen, by just having oxygen instead of nitrate, you double the amount of power of one NADH, which also shows you, why oxygen breathing organisms dominated organisms that didn't use oxygen, even though oxygen itself is a very toxic poison and it's super dangerous. When harnessed, it can create a lot more energy than something like a nitrate. So any questions about this type of problem? All right. So the last thing we're going to talk about today is we're just going to look at complex 1 to begin with. So remember, there are 4 complexes. Complex 1 is also known as NADH, coenzyme Q, oxidoreductase. So you can see why complex 1 is way easier to say. Here, we're taking electrons from NADH and we're passing them to coenzyme Q. This complex is huge. It is the biggest protein complex in the inner mitochondrial membrane. You can just see in the image on the bottom left there, right? Each one of these colors is a different protein. So all of these different proteins are coming together to make a massive complex. So 44 different subunits, mass of 980 kilodaltons. Yeah, that's ginormous. That's like a blue whale in terms of protein size. It looks like a giant L or an arm, and the lower part of the L or hockey stick or foot or whatever, or state of Florida, is in the cytoplasm, while the other part sticks out. And here we have one molecule of FMN, so flavin mononucleotide, as a prosthetic group. Remember, a prosthetic group is a non-protein part that is connected to an enzyme for it to work. And what happens is that NADH comes in and gives its electrons to FMN. So FNM is shown here, and it can accept 1 or 2 electrons. So NADH can only pass 2 electrons, so it gives it to FNN. Then, to get it all the way to coenzyme Q, we have these iron sulfur clusters. This is something that's going to keep reoccurring when we talk about ETC. To move electrons around safely, we have iron sulfur clusters where we have cysteines, which, remember, cysteines, the side chain is a sulfur, are connected to iron, which are connected to inorganic sulfur. So you can have a 2 iron 2 sulfur complex or a 4 iron 4 sulfur complex. And think of these as like little electron cages that we use to pass the electrons down in a stepwise manner to coenzyme Q. And what coenzyme Q will do is, again, it can accept 1 or 2 electrons at the time. So coenzyme Q with no electrons versus coenzyme Q with 2 electrons, we call that QH2 because it has 2 hydrogens with the electrons. If it just has 1 electron, it's QH, the radical form. So that is how we begin the electron transport chain for NADH going to UQ or coenzyme Q. And since it's the end of time, I'm going to leave the last part of complex 1 with how electron transport leads to proton pumping. I'll leave that to Tuesday. So that way at the very beginning of Tuesday, we can refresh on complex 1. And we will just go over that and continue with ETC. Right. So like I said, it's 1:45 now. So if you have any questions about anything we go over, please don't hesitate to email me. I'll do my best to answer those in a timely fashion. I'll put up homework so you can review what we learned today. See if you got the concepts down. Otherwise, I don't have anything else for you. I hope you all have a good weekend. It will actually looks like it's getting warm again. And hopefully I will see you on Tuesday. Have a good weekend, everybody.